Hey,
Hi,
how are you？
James.
你好，嘿，这个上一回我
Good
听你的，
morning。
在
彭飞。
西湖，对，Yeah，西湖大学做报告的时候，我听
哦，
，very good，very
哦，
good。
谢谢谢谢。我
嗯，
我英文不太好，
然后我
讲的可能不一
非常
定。
清楚，那天讲的非常清楚，你说话比较慢，比较像比较慢，但是很清楚。
哦，今天要讲肺，我因为是嗯 Chinese American Lung Association，所以我用英文讲。
Yeah，yes，yes，我们
啊
都是用英文讲的，对的，对的。
啊
因为有的时候我们也有些外国朋友来，
啊
嗯，
！Hi，金三。
你好，哎，你好，你好，Look forward to a talk，好长时间没见了，
你好。
哎，
你们都认识是吧？那就不用介绍。
认识，哎，周教授给我这边帮助很大的，非常感谢。
哦，对对对，我想起来，
对，我
他给的
们，
给的
对对对，
mouse 啊
那
，That's right。
个那个 Dream Mouse 就是给那边帮我设计的，works really good。
啊，
建文，我其实想我看看，最近有没有写些东西，有机会我看也把我们的工作也可以在这里跟大家分享一下，看看大家一些
好啊，好啊，
专家的
好啊，好啊，
一些反
好啊，
馈。
好啊，好
这个
啊，好啊，那给你安排一个 talk，那就
对，我觉得斌这块儿他不光是自己做得好，就是给很多人提供了非常 general 的 support，我觉得这点也是非常难得的。我是深有体会，非常感
对
谢啊！
对的。
回头我会把
不客气。
这工作跟你稍微聊一下，也
嗯，
是跟这个
我们
让 civil
我怎
center。
么可以知道CALA 的这些 seminar 的 information？就
呃
是如何 get 这种 link 以后，比如我
，yeah
也想
yeah
去
yeah
听。
，OK yeah，we will invite you. I don't know you. Do you have a WeChat? 金山 do you have
哦
a?
，WeChat yes。
So we have a group actually. So we could
那等下我发个邮件给您，我把我的 WeChat 的 ID，或者怎么能够加入这个
我有斌，我有你的，我帮你弄，我跟建文把这个视
啊，就
频。
是加入 CALA 的那个群里，这样我可以知道什么时候有报告了。我也想去学习，因为我其实只是对肺挺感兴趣，但我还是在学习过程中做
挺
肺。
好的，挺好的，Welcome。我们这个 community 大概有60-70个 PI，大部分在美国，有一部分在中国。哦，对了，我们6月底，6月28号到30号要在上海举办第二次的这个就是 CALA 的国际会议，Lung 会议，
哦，
欢迎
这叫
参加。
CALA 哦。
对
CALA CALA 啊。
，CALA yes，对。因为美国有很多这种，就在美国有好几个这种协会，比如说这么diabetes 的 DACA 吧，DACA
啊，
DAKA，
打卡哦。
对，还有 cancer 的也有一个这样的协会，中美 cancer，
打卡。
嗯就是 easy to remember。
嗯
我们再等1分钟，然后我们就开始。
好的。
对，我们把你加进群里以后，里边你就可以认识里边的，就是大家可以相互认识，做废的，在美国和中国主要
嗯
是中国人，Chinese scientist，我们可以合作。我知道你是一个很 collaborative 的人，也非常 honest，which I really appreciate。
然后因为主要 request 特别多，有时候我自己 handle 不过来，有时候会。
Yeah，不，但是 you are very responsive，very responsive 的。嗯 OK，maybe you can get started。
好，那我共享屏幕了。
Yeah，你先共享一屏，然后我介绍你。
Can you see the screen？
You are sharing. Yes.
Okay,
Yes.
looks good.
Yeah, full screen. Yes. Okay. So It's a great pleasure to introduce Dr. Bing Zhou, who has made great progress in building new tools for lineage tracing stem progenitor cells in multiple organs, including the lung. And Dr. Zhou is a professor and new Cornerstone investigator in Chinese Academy of Science in Shanghai. Just a little bit background of about Dr. Zhou. Dr. Zhou was trained as a MD, medical doctor in Zhejiang University School of Medicine, and then obtained PhD degree from Peking Union Medical College in 2006. And from 2006 to 2010, I think, Dr. Zhou did his postdoc training with William Pu or Bill Pu at Boston Children's Hospital, studying heart development in stem cells. And in 2010, Dr. Zhou started his lab in Chinese Academy of Science, where his science career really take off. Published very well in top journals, including Nature Genetics, Nature Cell Biology, very frequently, and very recently have a paper in Cell Stem Cell about lung, basal cells, I believe. And he has built several mouse lines that are widely used, including myself, in studying stem cells in different organs like liver and lung. And today, he's going to share with us his new findings in the lung. And I look forward to your talk, Dr. Zhou, and the floor is yours.
Thank you, Jianwen, for the kind invitation. It's my great honor to give a talk in CALA seminar. Today, I will introduce you the application of dual recombinases for study of lung stem cell and regeneration. At the painting this Paul Gauguin, there are several major questions in human life. Where do we come from? What are we? And where are we going? Similarly, in the multicellular organism, we are interested to address these three fundamental questions. What are the cell origins, cell identity, and cell fate? The method to delineate this process is called genetic lineage tracing, which take advantage of bacteriophage P1 Cre recombinase. Using Cre-LoxP recombination, we apply this in the genetic lineage tracing in mouse. We can understand the cell proliferation or cell differentiation. The working principle for Cre-LoxP lineage tracing is before Cre recombination, the reporter is blocked. The transcription of reporter is blocked by the stop cassette. After Cre-LoxP recombination, the stop will be removed, resulting in the reporter expression permanently in Cre-expressing cells and all the descendants. So we could permanently and heritably trace these Cre-positive cells and all descendants. By using single recombinase mediated lineage tracing, there are many controversies over cell differentiation and proliferation. For example, in the heart, there is controversy over the cardiac stem cells or cardiomyocyte is generated mainly by cell proliferation. There is also controversy in liver or in pancreas about the hepatocyte origin or beta cell origin. Do these cells proliferate by themselves or can be derived from progenitors? The reason for this controversy is largely due to single recombinase mediated lineage tracing because CRE is driven by a promoter. However, sometimes the promoter is not specific. To make lineage tracing more specific, my lab introduced a new recombinase to combine with the CRE-LOXP. It's called DRE-ROX. We can see that the CRE target on LOXP, but not on ROX. DRE target on ROX, but not LOXP. So CRE-LOXP and DRE-ROX are two orthogonal recombination systems. Taking advantage of these orthogonal characteristics of these two recombination systems, we apply them to perform lineage tracing with enhanced precision. So we apply this dual recombinases to multiple fields. Today I will introduce you the lung regeneration. We use these dual recombinases to study stem cells in lung regeneration. So my talk includes two parts. The first is bronchial alveolar stem cell or BASCs. The second is the origin of alveolar stem cell. It's alveolar type 2 stem cells. In 2005, Clark King first identified the bronchial alveolar stem cell BASCs, which reside in between the bronchioles and alveolar. These stem cells express markers of club cell and also 82 cells. However, the function or their cell fate in vivo remains controversial or remains largely unknown. So we aim to specifically genetically target this BASCs population. We trace BASCs using dual recombinase. As we know that the BASCs is sit in between the club cells and alveolar structure 82 cells. And these cells express both 82 cell marker, SFTPC or SPC, and the club cell marker, which is encoded by gene SCGB1A1. So we use dual recombinases. Specifically, DRE-ER is driven by SFTPC, which is 82 cell marker, and SCGB1A1, which is a club cell marker, drives CRE-ER. And we use ROSA 26, ROSA ROX stop ROX, LOXP stop LOXP TD tomato. After tamoxifen injection, the DRE-ER target on ROX flanked stop remove this first stop, and the CRE-ER also removes the LOXP flanked stop. So both stops are removed so that the this CC10 positive, SPC positive BASCs will express TD tomato. Because it's a permanent lineage tracing, all the descendants of these BASCs will be genetically marked. So in this way, we could label the BASCs that express both markers for 82 and club cells by TD tomato. So this is the data for BASCs tracer. We stained the BASCs tracer mouse lung by antibody CC10, which is a club cell marker. You can see that the TD tomato labeled cells are highly enriched in the end of bronchioles, which is at the bronchial alveolar duct junction. So magnification of these TD tomato positive cells will show they express the club cell marker CC10. We also stained the sections with the 82 cell marker SPC. And this TD tomato, which is located in the BADJ, also express 82 cell marker SPC, which is denoted by the arrowheads. Then we induce the mice with some injury models. We first use naphthalene, which induces the bronchial injury, and then we follow the cell fates for about 9 weeks. And we found that there are significant TD tomato positive cells in the naphthalene treated mice compared with the vehicle. Immunostaining for CC10 TD tomato on the naphthalene treated mice lung sections, we found that there are a substantial number of club cells express TD tomato, suggesting that these new club cells are derived from pre-existing basks. Compared with the vehicle, there are a significant increase of the TD tomato positive cells and CC10 positive cells in the naphthalene treated mice. So this data demonstrated that in the bronchial injury, basks could migrate into the bronchial and generate club cells and also ciliated cells. Then we treated the mice with another injury model, which is bleomycin-induced alveolar injury model. And after 8 weeks, we found that there is a significant increase of TD tomato signals in the lung compared in the bleomycin-treated lung compared with the vehicle. And we found that these TD tomato express SPC, which is 82 cell marker, and also some slender type long-shaped 81 cells. And the quantification data showed that there are insignificant increase of SPC positive, TD tomato positive cells in the bleomycin treated mice compared with the vehicle treated mice. So this data demonstrated that in the alveolar injury, basks respond to this type of injury and generate alveolar epithelial cells, including 81 and 82 cells. Next, we performed a clonal analysis of single basks because we want to understand if single basks can give rise to different types of lung epithelial cells. So we generate a confetti mice. We call this Rosa 26 confetti 2 because it responded to dual recombinases, which resulted in either RFP, YFP, or GFP in dual recombinases expressing basks. So in this 3 color, we could perform clonal analysis of single basks. After tamoxifen injection before injury, we could find sparse labeled single colored single individual basks. And after naphthalene induced bronchial injury, we found clone 1 and clone 2, which are shown in either GFP or RFP, which is single color, demonstrating that these cells are more likely to be derived from single basks. Next, we performed the bleomycin-induced injury and found that a cluster of cells which are derived from single basks appeared surrounding the BADJ, and the cell number is significantly higher than that of control mice. We also found that these cells differentiate into the 81, which is marked by T1 alpha. So this study demonstrated that we could use dual recombinase to specifically label a stem cell population like basks, which share 2 molecular markers. And we found that during homeostasis, these basks self-renew slowly. And under bronchial injury, they generate club cells and ciliated cells in the airway. And after alveolar injury, these cells contribute to AT1 and AT2 alveolar epithelial cells. So this data demonstrated that the BASEs are multipotent progenitor population for lung repair and regeneration. My second part of talk is introduce you an unpublished work about the origins of alveolar stem cells, AT2 cells. As you know that the AT2 cells is very important for alveolar maintenance and structure because they are the progenitor or stem cells for alveolar. They self-renew and also differentiate into AT1 cells. It is well known that AT2 cells produce by themselves through proliferation. However, other studies suggest they can also be derived from AT1. AT1, which is known for differentiated cells, which now is many studies suggest that AT1 can dedifferentiate and contribute to the AT2 cells in homeostasis, in diseased conditions, and even in tumor models. And recent studies suggest that the club cells, for example, which is marked by H2K1 high positive subpopulation of club cells, can migrate out of the bronchiole and contribute to AT2 cells after injury. And as I have just introduced to you that the BASEs can also contribute to AT2 cells. So there are many different sources for AT2 cells. However, most of these studies are done by single recombinase mediated lineage tracing. I just explain why single recombinase has some caveat in lineage tracing for study of AT2 because it's non-specific tracing. For example, most people use HOPAX to lineage trace AT1. We use HOPAX Cree-ER or Dray-ER and found consistently that these HOPAX positive cells are not only expressed in AT1 cells, but also in a large proportion of club cells and also ciliated cells. We use a different AT1 marker, which is AGER. We use AGER Cree, we found it is expressed in AT1. In addition, they also express in a small number of AT2 cells. So in this way, the HOPAX or AGER lineage tracing targeting on AT1 and actually not only AT1 cells could be labeled, but also other types of cells like club cells or a few number of AT2 cells are labeled. This would confound the interpretation of lineage tracing data because it labels other cell population that also contribute to AT2 cells. Then we also check the SCGB1A1, which is a marker for club cells, and SFTPC, which is a marker for AT2 cells. However, these two markers mediated single recombinase also target BASEs, as I have shown you that BASEs can contribute to AT2 cells. So these cells, when we interpret this data, we can overestimate their contribution to AT2 cells because they include the other cell population like BASEs that can also contribute to AT2. To circumvent this caveat, we generate a dual recombinases mediated lineage tracing for AT1. As I have shown you that HOPAX and AGER target on different population of epithelial cells in addition to AT1 AT1. We combine these two markers, and the intersectional part of these HOP-X and AGER actually can target more specifically the AT1 cell population. And when we perform lineage trace of dual recombinases, by dual recombinases, we found that they can specifically label AT1 cells without label AT2 club or ciliated or club cells. We generate this HOP-X, DREER, AGER, CREER, those are 26 ROX stop ROX, LOXP stop LOXP TD tomato. This upper panel is HOP-X CREER lineage trace. You can see that many TD tomato positive cells express SFTPC. However, HOP-X CREER also label a large portion of club cells, as we know that club cells can also contribute to AT2 cells after injury as they migrate out and differentiate into AT2. So this contribution could be overestimated. By using the lower part, the lower panel is dual recombinase mediated lineage tracing data. We found that these TD tomato positive cells, which is AT1 specifically labeled cells, they do not co-localize with SFTPC. We did not find any TD tomato positive cells, which are now AT1 specific labeled cells. They do not contribute to AT2. So this is under bleomycin injury. So we next change into a second injury model, which is pneumonectomy, PNX injury model, which has been reported that in this model, AT1 contribute to AT2. And we use HOP-X CREER on the upper panel of images. We found that these TD tomato positive cells label SFTBC. However, using dual recombinases, which are in the lower panel, these TD tomato positive cells are not co-localized with SFTPC. Here is the quantification data suggesting that under PNX injury model, AT1 cells do not revert back to AT2. They are fully differentiated cell type without contributing to AT2 cells during homeostasis or in the bleomycin injury model or in PNX injury model. We also tested in other models. We didn't see their contribution to AT2. Next, we want to address the exact contribution of AT2 cells, club cells, or BASEs to AT2 after injury. So we generate a model called triple light reporter, which has the LOXP stop LOXP, TD tomato, and ROX stop ROX, GFP. And we use SCGB1A1 CREER, which target on club cells and BASEs, SFTPC DREER, which target on AT2 and BASEs. And using this triple light reporter TLR, we could distinctly label 3 populations with 3 distinct colors simultaneously in single mouse. For the club cells, which are SCGB1A1 positive, but SFTPC negative, is shown by the red TD tomato color. And the AT2 is SCGB1A1 negative, SFTPC positive, is shown in the green GFP color, while their intersectional part is BASEs, which are double positive for SCGB1A1 and SFTPC, is shown in TD tomato and GFP. So in fluorescence, it's a yellow color. Here is a cartoon image showing that we could label the club cells, airway epithelial cells by TD tomato, BASEs by YFP, and 82 by GFP. So we compared the sham and the injury induced by bleomycin model. We found that under sham condition, the TD tomato are enriched in the club, while the GFP ZS green is in the AT2 cells, and the bronchial basics in the bronchial alveolar duct junction is YFP. So it's both TD tomato and GFP positive. After bleomycin injury, we found that a cluster of cells shown in yellow and also in TD tomato appearing in the area region close to the bronchioles, and they express SFTPC marker. The yellow arrowheads point to the yellow arrowheads point to the basics tracer, which are derived from basics, and they contribute to the AT2, while the white arrowheads denote the club tracer, and they are derived from club cells, and they now contribute to SFTPC. The right panel is the quantification data of the percentage of lineage traced AT2 cells. In the sham, compared with the sham, club tracer, basic tracer derived AT2 significantly increased. So this triple color linear tracing showed that after injury, the club cells and the basics could both contribute to the AT2 cells, in addition to the AT2 self-proliferation. As we see that the club cells can contribute to a portion, many AT2 cells, we want to address the question of the potential or capability of club cell to AT2 cell conversion under some extreme models. We performed 2 extreme models. One is ablator's AT2 cell, another is overexpression of P21 transcription factor that inhibits cell proliferation. So it impairs AT2 cells proliferation. So we generate a ROSA26 ROX stop ROX P21 GFP. And this is mediated by Dray ROX recombination to express the P21. We express the SFTPC Dray ER. So P21 would be expressed in the AT2 cells and the basics as well, because basics also express SFTPC. So basics and AT2 cells proliferation would be impaired by overexpression of P21. Then we want to test if these club cells can generate AT2 cells. To what extent can these club cells contribute to AT2 cells, as this AT2 and basic self-renew is impaired? Now, after 4 weeks of injury, we found that a larger cluster of TD tomato positive cells can be observed. These TD tomato positive cells express the SFTPC. Here is a quantification data showing that the club tracer can contribute to as many as 40% of AT2 cells after 4 weeks. Then we extend the time window to 8 months. Now we see that the majority of AT2 cells are now derived from TD tomato positive club tracer, which are originating from bronchial club cells. Now the club cells can contribute to as many as 80% of AT2 cells in some lobules of lung, suggesting that under special conditions, club cells have strong capacity to contribute differentiating into the 82 cells. Next, we want to address the molecular mechanism regulating different cell populations contribution to to 82 cells. We cross the triple light reporter mice with the RBPJ flux, which is a notch transcription factor, to knock out the notch signaling. We use the flux wild type as legitimate control, and because the SCGB1A1 CRIA target on both club cells and basks, so notch signaling would be knocked out in club cells and basks. Because we could distinctly label these two populations with two different fluorescence, so we could compare the impact of notch knockout to these two different subtype of cells in their contribution to 82. The upper panel is control mice, which is RBPJ flux wild type. We found that there is TD tomato cluster and a YFP cluster coming out after 4 weeks post bleomycin injury. After notch knockout, in the notch knockout mutant model, we found that the yellow part, which is a base tracer, the yellow cluster are significantly increased, while the TD tomato cluster are reduced. This is the magnification data showing these cells contribute to the SFTPC positives, class 82 cells. And the quantification data showed that after notch knockout, the contribution of club cell to 82 cells is significantly reduced. Instead, the contribution of bases to 82 cells are increased. Here is the quantification data by flow cytometry analysis, which confirmed the above immunostaining data, showing that club cells or bases, their contribution to 82 are distinctly regulated by notch signaling. And in between the club cells contribution to the 82 cells, we identify a transient or transitional progenitor population marked by P63. When we inject tamoxifen after bleomycin, we found that there is some epithelial cells that surrounding the bronchioles are labeled by P63 CRIA. These cells express P63. And then we follow their cell fate and found at 4, 8, or 20 weeks later, these cells differentiate into SPC positive or AGER positive 82 and 81 cells, suggesting that this injury-induced transient P63 positive progenitors contribute to the alveolar epithelial cells after injury and repair. Then we want to test if the club cells contribute to the P63, and then this P63 contribute to 82 cells. This sequential genetic lineage tracing, we generated a P63 LOXP stop LOXP delay, and after lineage CRIA like CC10 CRIA, you could excise this stop and make this P63 delay. And after injury, the P63 would be expressed in response to injury, and the delay would then target on ROX stop ROX and remove the next stop and turning this cell into TD tomato. Then this is the data showing that the club cell derived P63 derived 82 cells and 81 cells. they express SPC. This data demonstrate that the P63 sit in between the club cells and AT2 cells. They are transitional progenitor cell population. Next, we knock out P63 in the club cells and test if their contribution to alveolar repair or regeneration is impaired. We found that in the P63 flocks delta mice, there are significantly reduced TD tomato positive cells in the alveolar compared with the legitimate control, which is P63 flocks wild type. This data demonstrate that there is an injury-induced P63 population, which are derived from club cells, and they eventually contribute to alveolar regeneration. And this process is controlled by P63. So this study using dual recombinases showed that in the homeostasis or after injuries, the AT1 cells do not generate or contribute to the AT2 cells. AT2 cells can self-renew, and in addition, club cells and bases could also contribute to AT2 cells. Our study provided a quantitative data on the extent of their contribution simultaneously in the one mouse after injury. We found that in the notch knockout model, the notch knockout significantly impaired the contribution of club cells to AT2, but promote bases to AT2 contribution. And in between the club cells to AT2, there is an injury-induced or responsive P63 positive progenitor population. And if we knock out this P63, the contribution of club cells to AT2 cells is significantly reduced. So this is our work on the dual recombinases and its application for the study of lung stem cells and the regeneration. Finally, I'd like to thank my research groups and collaborators, and also from these fundings. Yeah, thank you for your attention. This is my talk.
Okay, well, wonderful. And now the floor is open to discussion and questions. And before you guys are preparing, I would like to ask a question as a host. So that's very, very interesting stuff, really. I like all the tools that you have built. But I was wondering, right? Please keep that picture here. So you have shown that SPC, when you do injury repair with bleomycin, there was a lot of SPC positive cells in terminal airways. You have shown with this dual 3 and CRE system. And
yeah.
if you, I do a lot of such experiments as well. If you can see a lot of cells in a terminal airways, so it's very difficult for me to say that how many cells are really only CC10 positive, but not the SPC and CC10 double positive. Do you know what I mean? Trying to say it's not only just limited to the terminal airways. It's actually, and not limited to those BADJs, but actually terminal airways. A lot of such cells are lining around that area.
Oh, yeah. Maybe after injury, the boundary between bronchial and alveolar may be not exact the BADJ. So there are many complex remodeling and complex things happening in this injured area. So many epithelial cells surrounding this area could express potentially express double markers, but our lineage tracing model is induced before injury, and if after injury they migrate out and spread out, so you can see a lot of TD tomato or yellow YFP positive cells. They are derived from club cells or basks, so they migrate out and contribute to the 82.
Okay.
I'm not sure if I have
No, yeah,
addressed that.
yeah, I understand that. Yeah, okay. We can discuss more later on. Yiru, you have a question?
Thank you. It's a great talk. So I have a question about this notch signaling. So I also know that we have seen publication several years ago, those basal cell-like cells, some keratin 5 positive, P63 positive cells can also become type 2 cells. And in their study, it seems that inhibit notch is required for those cells to become type 2 cells. But in yours, it seems notch block those cells to become type 2 cells. So how do you explain why there's a different mechanism?
Yeah, thank you. Yeah, we also see that paper. We don't know because we have different lineage tracing system. We originally thought we could recapitulate their data, but when we knock out the notch and distinctly label the club and basks, we didn't label the K5. Maybe in future we could use some dual recombinases to label basal cells and other cell population together and see if their contribution to 82 could be impaired in the notch knockout model. But in our study, as we distinctly distinguish club cells and basks, so we has its internal control. We found that the club cell contribution to 82 is reduced by notch, but the notch knockout promote the basks to 82 cells. Maybe it's, I think it's different system, and we are confident that our model more clearly sees the distinct subpopulation of cells. Yeah, that's what my explanation.
Yes, thank you. And I have another technical question. So we know that those P63 positive, keratin 5 positive cells can become type 2 cells. And I think when in the process of they becoming type 2 cells, I don't know if correct me if I remember wrong, but I remember sometime when they at the process that differentiating, they also sometimes transiently express CC10. So in your lineage tracing, I know you label those CC10 positive cells before the injury, but is it possible that some tamoxifen is still in the body and after the injury, some other cells start to acquire CC10 and then they were also labeled?
Oh, thank you for your question. I think in previous studies, they use K5 or P63. They use H1N1 virus infection model. Our model is bleomycin. We did the experiment of P63 Cree-ER lineage tracing by tamoxifen injection before injury. We did not see their contribution to 82 cells in bleomycin-induced injury model. But if we use virus infection model, we found they could contribute to some 82 cells. So we have different injury models. Our model is bleomycin-induced injury model, and we found only after the injury we inject the TAM, we can trace this transient population. And what our cells may be P63 different from the previously reported basal-like cells. They are two different type of cells. The previously labeled is K5 double positive, is basal-like cell. They contribute to bronchial-like structure, and which is regulated by hypoxia. And some reports suggested they do not differentiate fully into AT2, while our study is on the bleomycin injury model. And after injury, they appear, and they appear surrounding the terminal bronchial structure. They are derived from club cells, which migrate out to become AT2 cells. So we have different models, injury models, and the cell population we traced between ours and previous reports are also different.
Okay, thank you.
Thank
Okay.
you.
Xin has a question after Yulu, because she cannot raise her hand. So Xin, it's your turn.
Thank you, Jianwen. Bing, it's great to see you. Thank you for
Glad to see you.
really amazing talk. I love mouse genetics, so this is just really spectacular array of tools that you've generated.
Thank you.
So I have a experimental question and maybe a speculative. I would like to ask you to speculate. So the experimental question is, I was actually surprised that from club cells to AT2, it goes through P63. So, knowing that P63 is more the airway basal cells, I would have predicted, if I was going to bet money on it, I was predicted it's going to be the other way around. So my question to you is, have you tested the other way around? Do the DRE and CRE combinations to test whether P63 could be actually also going through some populations of it, could also going through club cells before becoming AT2.
Oh, that's a very good question. Yeah, if P63 could go through club cells to AT2, do you mean this P63 is what we mentioned, the injury-induced
Yes.
P63 expressing cells?
Yes.
Well, I haven't thought of this. It could also go through the club cells. Actually, we could use dual recombinase to address this question, as we are generating a mouse called CC10 SCGB1A1 LOXP stop LOXP DRE, and we want to address if some cells could go through the club cells as intermediate stage, then generate other type of cells. Yeah, we could use this dual recombinase to capture the transient club cell CC10 expressing cells using a kind of LOXP stop LOXP DRE system. Yeah, that's a very good suggestion. We would
you just have
in
to
future test it.
Induce it after the injury, right? So after the basal cells are there.
Yes. This is very cool idea, that if some cells can go through club cells to give rise to other type of cells as an intermediate state.
Right. I'm just wondering if the basals, the injury-reduced basal cells and club cells are equally residing on the more plastic stage. So, you can go through either way, sideways. It's a bidirectional thing.
Yeah, thank you for the suggestion. I mean, if we go through the club cells, it's the kinds of basal cell-like type of cells. But if it doesn't, it may be it's a transient population that derived from club cells to AT2 cells.
Sure. Yeah, so I wouldn't suggest it to anyone else, but I know you guys can do those amazing combinations.
Thanks for your wonderful suggestion.
So my question for you to speculate is, how much of this sort of plasticity is conserved in human based on what you have data for?
for human, we may not do the lineage tracing directly, but alternatively, we can use some organoid technology to test if in vitro this cell has some plasticity by lineage tracing of human cells, and then do single cell transcriptome and analyze their multipotency in vitro model. But in human beings, I think there is plasticity for the cells in between the alveolar structure and bronchial structure, as Tata in Duke University showed that in human, the AT2 cells has a plasticity. Alveolar stem cell has a plasticity to generate some club cells, which is very surprising. Although we haven't shown in mouse, maybe there is some plasticity, or maybe there is difference in between the human samples, humans and the mouse. But I think there should be some consistency in human and mouse for some stem cell population. But we also know that the structure of human and mouse is different in the terminal structure. So there's some plasticity of stem cell conservation, but we should also know there is a difference between the terminal structure. Maybe there are more differences or similarities that we needed to use better technology to understand in future.
Yeah, so Tian's lab has also, Tianpeng's lab has also shown that the AT2 cells in combination with the human mesenchymal cells can give rise to kind of basal cell looking structure. So there's also that precedence there. Yeah, so thank you very much.
Thank you, Xin.
Okay, our next question coming from Feng Yuan.
Hi, this is Feng Yuan from University of Taiwan.
Yeah,
The talk
nice
is
to meet you.
really, it's really good, and I think this is such a beautiful work. It really like answers some of the key important questions in the field. You know, in the long regeneration field, and it's super controversial on the origin of AT2 cells. Group of people, and they think AT2 cells are stem cells, but some of the people, and they think basal cells or distal alveolar basal cells can regenerate AT2 cells. However, this is very controversial. Those two group of people always fighting to each other, but this paper kind of satisfy both group, I think. So my question is about the P63 positive progenitors. How do you think that those are just alveolar basal cells, or they are different from alveolar basal cells?
I think they are different from basal cells. They are not previous sort of the like bronchioles, the basal bronchial cells, the basal cell in the bronchioles. They are rather a type of cells that sit in between the club cells and AT2 cells. They express club cell marker. They are different from previous K5 positive cells. They also they share some by single cell transcriptome analysis. We found that they share some gene markers with K5, but not quite overlay. So they are distinct subpopulation that I think is more, we explain it is in injury induced P63 positive cells that are derived from club cells. We have genetic evidence to show that they are derived from club cells, and they differentiate into AT2 and AT1. So I think they are different from our conventional defined basal cell expressing K5 or P63.
Yep. So one more question. So do you think the club cells go into the P63 positive cells are like a style of de-differentiation? Like, so because we think the P63 positive basal cells, they normally can generate CAR cells in our during our regeneration. So I think this is a like kind of de-differentiation in the process, like the
yeah. Thank you for your question.
Goes back to the progenitor state.
Yeah, it's a good idea that this. I mean, we know that the basal cell gives rise to club cells, and the club cell now express the conventional basal cell marker P63. It's kind of de-differentiated to its parental cell, but they are not exactly the same as basal cell. In addition, we found that only about half of the club cell undergo this P63 pathway or express P63. It's not due to efficiency, it's because we use P63 LOXP stop LOXP delay. So any cells that go through the P63 would be genetically tagged. We found that only half of the club cells that contribute to 82 cells go through this pathway. So that means another half, they do not express P63. So they maybe go through other pathways to contribute to 82 cells. So I think these club cells may transiently express maybe, as you call the de-differentiation of their parental cell transcription factors, but they are not the same. Yeah, that's my explanation.
Okay.
Thank you.
Could I just do one more question? So most of your work are focused on the major cell types. Have you ever looked at rare cell types, rare epithelial cells like neuroendocrine cells? Like, for example, are we able to say this kind of CAR cell lineage goes to the neuroendocrine cells during injury?
I haven't go in details for the neuroendocrine cell. I think Xin and Penfei has done a lot in this field, some very great works. I am more focused on the terminal structure, like the picture shows, that the bronchial region, which is terminal structure, and the alveolar structure. We haven't checked in the neuroendocrine cells. Maybe it requires some very special model to induce injury in special regions that to see the lineage conversion for these neuroendocrine cells. Yeah, thank you for your suggestion.
Hypoxia will do it. You mentioned hypoxia earlier.
Okay, hypoxia.
Increase in health do it.
Thank you.
Yeah, thank you so much. It's great work.
Okay,
Thanks.
our next question from Jingsan Zhang.
Thank you, Chairman. I mean, it's such a wonderful talk. I know Bin is a hard person, not a long person, but the talk is so informed. I'm sure every one of us will learn something from this beautiful tools. Indeed, actually, being single-handedly, almost single-handedly build all the three toolbox. And I personally get the support from him for designing the PD-L1 dream mice for us. For the time's sake, I just have a quick like a question, because all these results, have you tried to use some of the single cell analysis combined with these models or the cell you identified? Because I think that should give lots of the more information regarding at the molecular level how these cells emerge or transdifferentiate. It can be technically challenging given the smaller number of these populations. That's something in my lab, we spent like a year to isolate some population. It worked out eventually. I'm just wondering if you have something at the single cell level to share with other people. I think we should be very interested to know something about it. Thank you, Bin. It's really a good talk.
Yeah, thank you, Jing-san, for the great question that about the single cell analysis. Yes, we do have did some single cell analysis of all these populations. For example, when we use triple light reporter, we isolate a basics tracer, club tracer, and 82 tracer, and compare their transcription and identify some transient population and compare them. Yes, we found many new findings through the single cell lineage tracing. If we want to know the transient subpopulation more definitively, we needed to do lineage tracing by dual recombinases for more definitive if one cell goes through this transient subtype of population to generate AT2. We have some single data showing this process, but it's a transcriptome, which is by calculation. If we want to definitively address this transient population, we can pursue in future by genetic study. Thank you for your suggestion. We are very interested in lung, and I'm still learning in this field. Thank you.
Thank you, Bian.
So next question is coming from Liu Gang, and he was asking in a chat box, and he was asking, have you tried to use hypoxia model that Morrissey and Nan
I did that.
Use. So what is your
didn't see
finding?
it.
I have that question as well.
Ed Morrissey has done a lot for AT1 transition to AT2 in UPenn, and we performed as many experiments as possible to try to recapitulate. But we found HOPX do contribute to AT2 cells, HOPX lineage, but by dual recombinase. When we use intersectional genetics, we found that under hypoxia condition, AT1 do not contribute to AT2. That's what we found. I haven't shown that today, but we didn't see it.
Okay. All right. Okay. Our next question is coming from Changfu Yao. Changfu? Are you muting or what? Hello? All right. Okay, I'm gonna skip you then. Our next question coming from Yan Hu.
Hi, Dr. Zhou. My name is Yan. I'm a postdoc fellow at the University of Colorado, about to be independent PI soon. So I'm really interested in your transgenic model to study the club cells and their progenitor cells. So it's exciting to see that not just the BASC cells contribute to AT2 cells. It seems like the club cells localized in the distal airway also contribute to AT2 cells. That's what I actually see a lot. So I've been having this question for a long time. So since they do both contribute to AT2 cells, are they transcriptionally similar, or what's the key difference between these two? And another question is, do they have differential responses to different type of injuries? So far I've been seeing that the injury model you have been using is the bleomycin model. So I wonder if you have tested, or are you interested in another injury model? So I personally are interested in a CUPD, so I'm looking at the emphysema development. So I'm really curious about how club cells contribute to the pathogenesis and whether they do tend to repair AT2 cell, this kind of non-stoppable alveolar destruction process. So maybe I would just let you answer if the club and the BASC cells are they really similar or not? Probably you already have some answer from your single cell RNA sequencing data.
Thank you for your question about the differentiation between the BASCs or club cells. Are they just the same population express gene differently, or functionally are they different population? This is a very important question. Before we knock out notch, could we know this? Because we thought it could be the different gene expression, but when we knock out notch, to our surprise, we can lineage trace them different color, and to our surprise, notch distinctly regulated these two populations. This is for the first time demonstrating that functionally they are two subtype of cells, not only for the molecular distinct, but also for the functional difference, because they respond differently after the same injury. So I think, personally, I think they are different functional population depending on the injury model and the genes you manipulate, try to dissect their difference under special conditions. They both can contribute to AT2. They are the same, they have the same function, but they are regulated by distinct molecular pathways or distinctly by notch pathways. So in future, one key technical limit for our study of basics is we do not have a specific tool to genetically delete genes in the basics. This is a hurdle for us, but now my lab has generated a new system that circumvent this hurdle. We generate a new mouse which can delete genes specifically in basics. So we could, in future, compare this with the club cells more easily. So I think it's a technical hurdle that make us less understand this basics population, because now we only lineage trace, we cannot manipulate them. But we now generate a new version. We use triple recombinases that can very specific, efficiently delete genes in that very small population. And then we could address the functional, because functional, it depends on the signaling pathway, not only the lineage cell fate, but also the molecular regulation for basics. So I think in future, we are happy to share all these reagents and to study like what you are interested, like COPD. We think only we have the tool to knock out the gene in basics specifically, can we address more questions and know this population better? Yeah, that's my answer. Thank you.
Thank you. I guess the next conundrum is to how to distinguish the club-only cells from proximal versus distal. I don't know if you already find the best marker.
Yeah, if we have good markers, my lab can help in making some genetic tools for all of you, but I don't know the good markers, like H2K1, but we haven't tried that.
Okay, thank you.
Thank you.
Our next question from Changfu, and his mic doesn't work. So he has a technical question. He was asking about P63 Cree mouse line that you made. Is it delta N form or TA form or pen P63 Cree?
I think it's we made many mice. I haven't. It's 2A. Maybe it's 2A Cree. P2A.
I'm sorry. No, I think no. I guess the question is that P63 has a major isoform.
It's germline. It's knockout. It's not a delta P60. Sorry, I misunderstood your question. Our knockout is P63 knockout, not a delta version.
I see. He was also asking about this P63 Cree. Do you target to delta and P63 Cree promoter or TA form? The promoter.
Promoter. Yeah, not a delta form.
I see.
We target on the promoter P63.
Okay. He also have another question. It's about the P21 overexpression mouse line. Did you see P21 overexpression mediated senescence?
That's a very good question. People thought P21 overexpression is senescence. We thought that, but when we check, it's another project. My student overexpressed P21, and she told me that these cells do not proliferate, but they are not exactly the same as senescent cell because they don't secrete SASP, these markers. So they are different. So about the senescence markers, whether P21 or P16 4A, which is a better one, or which is the best, more authentic senescence marker, we don't know. Maybe overexpression, P21 will only use it to impair the self-proliferation, but they are not the same as senescence cells, because senescence cells not only are not proliferate this feature, but also it should have other features, like one very key important feature is SASP. But when we overexpress P21, they do not express many these SASP markers. Yeah, that's my answer.
Okay, wonderful. Do we have more questions? Oh, yeah, Xiaoqing Wei.
Yeah, thank you. So I just have three sample questions. So the first is why you just go to using this? Actually, you have a very nice tool. It's very cool. So why just check the 82 cells? Why not also check the 81 cells? Because also the paper show different the source of 81 cells after injury. I just curious. Because 82 cells can come and 82. So I'm also curious during the injury, what other cells can directly go to the 81 or
yeah, that's a very good question. Yeah, we are trying to address this is a very important question. If any cell can go to 81 without the step of 82. The reason why we first started 82 is we know that 82 is a very central important of stem cell for the alveolar, because 82 generate 81. That's what we know. But for your question, if any cell can go to 81 without 82, this is very hard to address, because you can use a single cell to suggest that, but you need direct genetic evidence that it does not go through 81 to go 82 to go to 81. So we needed to generate some tools, which we are working on. If any cell that contribute to 81 without going step of 82, so this need a genetic study, which is now we could not definitively adjust, because a single cell you can suggest they do not they have different pseudo time. You can analyze that they go through an independent path without going through 82, but this is not definitive evidence. I think it could be possible that some cells can generate 81 without going through 82 directly. Yeah,
thank you.
It's possible.
Okay, thank you. And also, I'm curious, is it possible to also inject the pregnant mouse at when the mouse pregnant, and then check how, and also at different time points after the pup's born, and then how tracing how this long development during this process?
Yeah, sure. We did a lot for embryonic study for some heart development, and we inject tamoxifen to pregnant mouse at like E 9.5, 16.5, whatever. You can do that, but the more you inject, the more likely the mouse will get abortion. So you need to control the tamoxifen, and also there is some way to circumvent this abortion, or after birth, you can use pseudo mother to circumvent this technical problem, because some mice cannot give rise to new pups because of tamoxifen. You need to use a pseudo mother for breeding of these pups. Yeah, there's some way to do.
Yeah, I just curious why some cells they already decide can go to some way. I just maybe is and also their specific location. So I want to know when they have this decision, like which stage, which age they already have this fate. I just feel this. And also, do you consider to using the tumor genesis, maybe the lung cancer?
For what? Oh, yeah.
Which cell from can cause the tumor, like? Because still a question, right? Which cell, which source is the can cause to the tumor?
Yeah, that's a very good question, because I'm very interested in development and regeneration. My lab has done very few in tumors. My colleague in our institute, Hong Bingji, did a lot for tumor. But I noticed that there's a recent study showing that AT1 cell give rise to tumors by lineage using lineage tracing of using HOPX from a Stanford group, I guess. So they suggest it's a new source of a differentiated cells that can generate tumors, which is a surprise. But my lab hasn't worked on tumor, but it's possible that we can use HOPX, DREA, and AGER, CREA to do intersectional lineage tracing for AT1 and follow its cell fate to tumor. But we needed to iterate the system because KRAS, we needed to use a singular recombinase readout by sequential genetic lineage tracing to express KRAS. That we needed to modify the system to readdress the question. Yeah, it's an interesting question for how about the tumor, the origin of tumors. It's also about the lineage. Yeah, thank you for your suggestion.
Yeah, very cool. Thank you. We learn a lot. Thank you. And we're looking for, and also curious about your future work about this lineage tracing in the human organoid. Thank you.
All right. Okay. Our next question is coming from Yinsan. You're muted.
Hello, Dr. Zhou. Thank you for your talk. So I'm postdoc from Jianwen's lab. So I have a question follow Changfu's question. So overexpression, you said overexpression can just affect the proliferation. So I just curious if any lung injury, because I saw at 8 months there are a lot of club cell lineage cells in the lung. So why so many lineage cells in lung? And any type pre-existing type 2 cell be killed or just
Thank you. Yeah, thank you for your question. Yeah, it's a very good point. Why after 8 months the club cells and its derivatives populate the entire lung lobule in some regions? Because this is what I explained. In the injury, a lot of AT2 cells are died in the bleomycin injury. They have to proliferate if the pre-existing, the remaining AT2 cells needed to proliferate to compensate for the loss. But as we impaired their proliferation, they can't proliferate by themselves. So the few, maybe 20% of AT2 cells that derived from club cells now have the advantage to proliferate over the other pre-existing AT2 cells that are impaired. So by compensation and competition, over 8 months, they gradually populate and win over most areas over these pre-existing AT2 cells that are non-proliferative. So I think in another way, the AT2 cells are non-proliferative. They are maybe more apoptosis, and the club cells derived AT2 cells are normal. They can proliferate, so they could have win over the other by cell-cell competition. So I think over the long time window we give to the mouse, the more likely the club cells populate more regions of the AT2, alveolar region. So that's my explanation. Thanks for the question.
I follow up on Yinsan's question, but I thought your P21 overexpressions depend on tamoxifen, is it right?
Yes.
But then they must have some recombined type 2 cells they can survive in without the P21 overexpression, right?
Yeah, it's possible, because the recombination cannot always 100%, but SPC DREA is a very strong, for unknown reason, SPC is a very strong promoter in our hands always. SPC, very strong. So their recombination efficiency is very high. Maybe it's not 100%, maybe it's 90, 97, 98, 99. But it's possible some remaining 82 cells, very few percent, like 1%, they are not recombined. They have ability to proliferate. So in our 8 months, we can see about 80% of 82 cells are derived from club cells. Maybe some remaining part, as Jevon suggested, could be unrecombined cells that they could still survive because they have ability to proliferate. It's possible, yes.
Okay, well, one last question from Liu Xue. Why not you to ask yourself? If not, then I can convey the message. So he was asking, is it possible that you can overexpress P63 or notch to promote airway cells contribute to 82 regeneration and to prevent fibrosis?
Okay, thank you for the question. We have done such experiments to overexpress notch in the basics. It's instead generate some tumor-like structure. We overexpress RBPJ, so too much dose, high dose is not good. And for P63, we haven't such mouse. And yeah, thanks for your suggestion. And in future, we might to overexpress P63, but in a controlled manner, like a theta regulatory system, not to overexpress too many, which may not be good to the epithelial cell. Maybe they can become over-proliferate or become tumor. So it's a good suggestion for us. Thank you.
All right. Okay. If there's
Sorry.
Thank you
Is
for all
that-
the questions.
anyone want to ask a question? Oh, hey.
Yeah.
Hey, Xiaobo.
Sorry. It's good to see you being again. I was just kind of general question for the regeneration. How many of these 81 cells from the initial 82 cell, and how much contribution from club-derived 82 cell to the type 1 cell for the regeneration?
Oh, thank you for your question. We haven't focused much on the 81 cell quantification. We more focus on the 82 cells. Yeah, that's a good question. If the 82 cells derived from club cells or from 82 cells through proliferation has the same ability to generate their 81 daughter cells. We haven't, yeah, that's a good question. If their ability to generate 81 are the same or different, we haven't addressed that question. But that's a very good, yeah, question. We needed to check if the 81 are different. Thank you for the suggestion.
Yeah, the end is like 81 form the major structure, and the 82 cell number probably be smaller, like in the end of the homeostatic after the long recover. But it's very really interesting story.
Yeah,
beautiful
thank you
tools.
for the question. Yeah, we thought that the 82 cells, once they are 82, they should be similar, but we haven't tested this experimentally to test if these 82 cells derived from different sources have the same ability or proportion to generate new 81 cells after injury. Yeah, that's a good suggestion. Thank you.
Have a beautiful system. It's more proportion, like in the end they do the mice actually need this newly generated club cell derived 82 to repair the lung. Actually, the initial 82 stem cell, they will do the majority work. Just kind of curious for the
we can do that. First, we can quantify. Second, we can do secondary injury to see if the 82 cells from different sources, we know by their fluorescent markers, and see after secondary injury if their contribution to 81 cells are different or not. Yeah, thanks
right.
for the suggestion.
Thank you. Thanks.
Thanks.
Can I discuss another point? So I just reminded that based on the 81, 82 cells, this research, we know this transitional cells accumulation can cause the pathogenic. Do you think maybe join your club cells to the P63 positive cells, then go to the 82 cells? Some signaling pathway maybe will upregulation, join your club cells to P63 cells. Maybe I don't know whether I1 beta based on the previous paper showed those transitional cells, I1 beta can be upregulation in this KRT8 transitional cells. So I don't know whether your this I1 beta can contribute to your P63. And when P63 go to the 82 cells and there's some signaling, they need to downregulation to make they can successful go to the 82. If some signaling pathway they are overexpression or inhibited, then your P63 will accumulate in the lung. Whether this P63 progenitor cells accumulation can cause the pathogenic in the lung, I don't know. I just think those questions.
Oh, thanks, Xiaoqing, for your nice question. We will check if this IL-1 beta, as you suggested, or other pathways downregulated in P63. And we may use P63 to knock out one or two genes, the key genes you suggested, to see if their contribution to the alveolar epithelial cells are impaired or increased by these signaling pathways. Yeah, thank you.
Thank you so much.
It's wonderful, wonderful. It's wonderful discussion and wonderful presentation. And thank you so much. And it's been late here, but well, thank you so much. And we will look forward to our next presentation. And good night, everyone.
Thank you all for the
Thank
nice
you, Ben.
questions. Thank you, Xin. Thank you,
Yeah,
Jian,
good to
for
see you.
organization. See you later. See you.
Bye,
Bye-bye.
Jian.
Yeah, bye-bye. Bye-bye.
Bye bye. Good talk.